Welcome to the PubMed Daily Review for May 2nd, 2026. Today we scanned 5,854 new open access papers. Here are the top 5 selected for detailed analysis. Up next, our first paper out of 5. Artificial Intelligence. Enhanced Wearable Technology Enables Ventricular Arrhythmia Prediction by Martin Z. H. Koch and Fleur V. Tian. Let's break down its key contributions. Ventricular arrhythmias are a serious concern, often leading to sudden cardiac death. While we know that physical activity levels can be a risk factor, the way our bodies behave changes over time, and these changes might be crucial for predicting these events. This study explores whether continuous monitoring of everyday physical behaviors using wearable devices can help us predict the risk of ventricular arrhythmias. Researchers enrolled 303 patients who already had an implantable cardiovertifibrillator. For a full year, these patients wore accelerometers to continuously track their physical behavior. The study focused on identifying patterns in this data, like activity levels and sleep, and then linked these patterns to whether or not a patient experienced a ventricular arrhythmia requiring treatment. The study found that reduced daily physical activity and low day-to-day variation in sleep were associated with a higher risk of ventricular arrhythmia. Importantly, when artificial intelligence, specifically deep representations from neural networks, was used to analyze the behavioral data, the predictive performance was significantly better. The area under the receiver operating characteristic curve was 0.74, compared to 0.67 for traditional statistical methods, with a p-value of 0.05 for the comparison. This research demonstrates a promising new avenue for predicting ventricular arrhythmias. By leveraging wearable technology and advanced AI, we can identify individuals at higher risk before an event occurs. This opens the door for more proactive patient management and potentially reduces the incidence of sudden cardiac death. Future work will likely focus on refining these AI models and integrating them into clinical practice. Up next, our second paper out of 5. Genome-scale CRISPR screens identify actin-capping proteins as key modulators of therapeutic responses to radiation and immunotherapy by Sean Xian and Nipun Varma. Let's break down its key contributions. Radiotherapy is a vital tool in cancer treatment, used for over half of all patients. Excitingly, radiation can actually boost anti-tumor immunity, making it a great partner for immune checkpoint inhibitors. But we don't fully understand which genetic factors within tumors dictate how well this powerful combination therapy works. This study aimed to uncover those critical genetic players. To find the genes that matter, the researchers used a powerful technique called genome-scale CRISPR screening. They tested melanoma cells under conditions mimicking combined radiation and immunotherapy. By looking at which gene knockouts helped cancer cells survive, they pinpointed 2 actin-capping proteins, CAPSA3 and CAPC, as critical players. Their work revealed a complex mechanism where these proteins affect DNA repair and immune signaling. The screens revealed that knocking out CAPSA3 and CAPC actually helped melanoma cells survive radiation and immunotherapy. While this impairment of DNA repair made cells more sensitive to radiation and chemotherapy, it also triggered the STING pathway and increased a molecule called CEACAM1. This led to cancer cells becoming resistant to being killed by CD8 T cells. Interestingly, patient data showed that mutations in these genes were linked to higher mutational burden and a positive correlation with immunotherapy response. This study highlights the crucial role of actin-capping proteins in DNA repair and their surprising impact on how tumors respond to radiation and immunotherapy. While inactivating these proteins makes cancer cells vulnerable to DNA damage, it also helps them hide from the immune system. These findings open up new avenues for developing smarter combination therapies by potentially targeting DNA repair or modulating the function of these actin-binding proteins. Up next, our third paper out of 5. Real-time NIR-II fluorescence imaging-guided precision thrombolysis with a molecularly planarized phototheranostics agent by Guoxing Zhang and Haoming Wang from a Jiangxi Provincial Key Laboratory of Functional Crystalline Materials Chemistry, Nanchang 330031, China. Let's break down its key contributions. Thrombosis is a deadly condition, responsible for millions of deaths worldwide. Current treatments, like systemic drugs, are often imprecise, leading to significant bleeding risks. We also lack the imaging technology to accurately guide these therapies in real time. This gap highlights the urgent need for integrated solutions that combine high-resolution imaging with targeted, safe treatment. To overcome these limitations, researchers developed a novel molecular design strategy called molecular planarization engineering. They synthesized a new squarane dye, SQ-by-pH, and encapsulated it into nanoparticles. These nanoparticles, SQ-by-pH NPs, are designed to emit light in the NIR-II window for high-resolution imaging and generate heat upon laser irradiation for targeted therapy. This approach aims to create a single agent capable of both visualizing and treating thrombi precisely. In a mouse model of venous thrombosis, SQ-by-pH NPs successfully visualized the thrombus using NIR-II fluorescence imaging. Upon irradiation with a 1064 nanometers laser, these nanoparticles efficiently generated heat, leading to precise thrombolysis and vascular recanalization. The entire process was monitored in real time, demonstrating high-contrast imaging and effective therapy. Importantly, the agent showed low cytotoxicity and excellent hemocompatibility, suggesting a favorable safety profile. This study presents a significant advancement in theranostic agents. The molecular planarization strategy successfully created an agent that excels in both NIR-II imaging and photothermal therapy. SQ-by-pH NPs provide a compelling integrated platform for precise thrombolysis, addressing key limitations of current approaches. While promising, further research is essential to explore its potential for clinical application and ensure its long-term safety. Up next, our fourth paper out of 5. A opioid receptor superagonist analgesic with minimal adverse effects by Juan L. Gomez and Michael Michaelides from Imaging and Molecular Neuropsychopharmacology Section, National Institute on Drug Abuse Intramural Research Program, Baltimore, Maryland, USA. Let's break down its key contributions. Developing safe and effective pain medications is a major challenge. While opioid agonists are crucial for pain relief, their high efficacy often leads to dangerous side effects like respiratory depression and addiction. Researchers have tried to create safer opioids by reducing their efficacy or altering their signaling pathways. This study investigates a novel approach, a superagonist with a unique pharmacological profile that might offer potent analgesia without the usual drawbacks. To explore this, the researchers synthesized and characterized a novel compound called N-acetylfluornitrazine, or DFNZ. They used a variety of techniques, including binding assays and advanced imaging like cryo-EM, to understand how DFNZ interacts with the µ-opioid receptor. The study then moved to rodent models to assess DFNZ's analgesic effects and its safety profile, specifically looking for common opioid side effects. The results were quite promising. DFNZ showed potent analgesic effects in rodent models, with an ED50 of approximately 0.3 milligrams per kilogram. Crucially, the dose required to induce side effects like catalepsy was about 3 times higher. Even more encouraging, DFNZ did not cause respiratory depression or tolerance, even with repeated administration. Its reinforcing effects were also weaker compared to other MOR agonists. This study challenges the long-held belief that high-efficacy MOR agonists are inherently unsafe. DFNZ's unique pharmacological profile, including its signaling bias and limited brain penetration, appears to decouple analgesia from severe adverse effects. While these findings are from preclinical studies, they offer significant hope for developing safer and more effective treatments for pain and opioid addiction. Further investigation in human trials will be essential to validate this potential. Up next, our fifth paper out of 5. A multi-omics PTM atlas reveals key insights into metabolic reprogramming in colorectal cancer by Tian Yuan Li and Yong Dai from School of Medicine, Anhui University of Science and Technology, Huainan, PR China. Let's break down its key contributions. Colorectal cancer remains a significant global health challenge, and despite advances, effective targeted therapies are still lacking. A fundamental characteristic of cancer cells is their altered metabolism, a process known as metabolic reprogramming. While we know that post-translational modifications, or PTMs, are crucial regulators of protein function, their specific role in driving metabolic changes within colorectal cancer cells hasn't been fully elucidated. This research seeks to fill that gap by creating a detailed map of these modifications to understand their influence on cancer metabolism. To investigate the role of PTMs in CRC metabolism, The researchers analyzed paired tumor and normal tissue samples from 8 patients. They employed high-resolution mass spectrometry to comprehensively profile 3 key types of post-translational modifications: phosphorylation, ubiquitination, and malonylation. By identifying differential modification patterns and analyzing protein interactions, they aimed to pinpoint the specific enzymes and pathways affected. This multi-omics approach allowed them to build a detailed picture of how these modifications contribute to the metabolic landscape of colorectal cancer. The analysis revealed a substantial number of altered post-translational modification sites in colorectal cancer tissues compared to normal tissues. Specifically, they identified 59 differential phosphorylation sites, 263 ubiquitination sites, and 64 malonylation sites. These modifications were found on critical metabolic enzymes like IDH1, LDHA, PDHA1, and GAPDH, which are central to cellular energy production. Notably, ubiquitination showed the widest range of changes, indicating highly heterogeneous regulation, while malonylation was significantly downregulated overall. These findings strongly suggest that PTMs play a crucial role in reprogramming cancer cell metabolism. In conclusion, this study demonstrates that post-translational modifications are key players in the metabolic reprogramming scene in colorectal cancer by directly affecting crucial metabolic enzymes. The comprehensive atlas generated offers valuable new insights into the complex metabolic environment of CRC. These findings open up exciting possibilities for developing novel, targeted therapies that specifically address these PTM-driven metabolic alterations, potentially improving treatment outcomes for patients. That's all for today's PubMed Daily. If you found this helpful, please leave a comment, and subscribe for daily paper summaries. See you tomorrow.